Thank you for the opportunity. And my presentation topic and the focus for this presentation is going to be the work that I have been doing for past 1.5 years in my current lab. So we work on our research focuses on cardiovascular disorders. And as we know that cardiovascular disorders are the major number one life-threatening condition across the globe, and they are the number one killer. So the preventing cardiovascular disorder is one of the major focus of enhancing the longevity and the quality of life. So cardiovascular disorders, they manifest as atherosclerotic plaques, calcified arteries, cardiac hypertrophy, peripheral artery disease, myocardial ischemia, and fibrosis and stroke. The primary reasons for cardiovascular disorders have been identified as aging, diabetes, obesity, and chronic kidney disorders. So they all converge at accumulation of senescent cells and the SASP. So now there are various reasons for accumulation of senescent cells. There are various theories that have been put behind it. As one of the previous speakers said, that the healthy mind and how brain, your thinking, your consciousness affects your bodily functions, and your other the coexistence of diabetes and chronic kidney disorders. They all lead to the elastin degradation. Elastin, which is the part of which is very much the structural component of heart and the vessels, heart and vessels. So once the elastin starts degrading, it exposes the vascular smooth muscle cells, and also it gives the opportunity for all the mineral disorders. They give the opportunity for the phenotypic changes and the phenotypic switches to happen, and then the vascular smooth muscle cells, the osteoblastic genes, they are turned on, and they change their phenotype to bone-like cells. So there's a lot of apoptosis that is going on, and those apoptotic bodies, they provide the breeding ground for the minerals in the body, like the phosphorus and calcium, to deposit. And these calcified arteries, they are hard. They lose their elasticity, not able to perform their function properly, giving rise to all kind of complications that I mentioned earlier, like hypertrophied heart and myocardial ischemia, loss of blood supply. Even in case of atherosclerotic plaques that block the arteries, there is a deposition of the calcium at the core, and the vascular smooth muscle cells, they lose their tensile strength, their elasticity to stretch, and these senescent cells that are quiescent by nature, they are not cleared, which could be due to the loss of endogenous immune surveillance, and they secrete these exosomes that are very typical of the senescent cells and is known as SASP. The senescent phenotypes, and this consists of all kinds of cytokines, majorly IL-6, IL-1 beta, TNF alpha, and they are all pro-inflammatory, and they cause changes in the vessels. So the potential solutions that have been suggested by the groups who have been studying senescent cells for quite a long time, like the James Kirkland group and the Judith Campesi group, their research has shown that Senolytics, they decrease the plaque size when it comes to atherosclerotic artery deposits and artery disorders. And so the agents that they have tried are the Senolytics that induce the apoptosis of the senescent cells, and those are ABT263, dasatinib, and quercetin in combination, ABT767, FOXO4 interacting peptides, and a prototype drug Galser that are all senolytics, and they induce apoptosis of senescent cells that are deposited in the coronary artery, and they have been found beneficial. So, but still the clinical studies are lacking in the field, and there is only preclinical data that is available for this. So these drugs have been very effective in treating myocardial ischemia, where there is a necrotic core, or at a focal point, there is a deposition of all the senescent cells, but not so much for the coronary artery disorder or for the calcified arteries. So another approach that we are trying is to use the agents that decrease the SASP, that is the secretory phenotype, which majorly consists of the pro-inflammatory cytokines and growth factors that is responsible for the remodeling of the aorta, and which the remodeling of the aorta. So atherosclerosis and calcified arteries, these are one of the leading cause of the cardiovascular events. And the challenges with the senolytics in the coronary artery for treating coronary artery diseases is that it may cause heavy apoptosis, and the apoptosis has been suggested as one of the leading event that promotes calcification, since apoptotic bodies, they provide the platform for the calcific changes. And then there is extensive apoptosis also leads in case of increase of atherosclerotic plaque. It can lead to plaque instability. It can cause acute coronary syndrome and carotid artery thrombosis. So senolytics in this case can have are somewhat difficult to translate when it comes to clinical studies, whereas Senomorphics that primarily act on decreasing the SASP are relatively easier to move forward with, and they could provide possible solution to treat cardiovascular disorders. They do not cause they do not induce the apoptosis of the senescent cells. However, they decrease the SASP, which is also responsible for the paracrine senescence, the spread of the senescence to the healthy cells, and thereby gives the opportunity for the existing cells to remodel, and therefore are the preferred solution. So in case of aneurysm also, senescent cells, they accumulate at the site of bifurcation. And if we use senolytics in that case, then it may cause a massive rupture of the rupture at the site of bifurcation and can lead to sudden death. And therefore, our group is working on Senomorphics as the potential approach to treat calcified coronary artery disorders. So our current research focus is on identifying the target and the source of SASP in medial artery calcification. So the model system that we use for this is the cell cultures. We use human primary vascular smooth muscle cells. We use primary endothelial cells. We use immortalized human aortic vascular smooth muscle cells to study the effect on the phenotype of various environments like high phosphate conditions, what happens to, how do the cells behave when we add angiotensin to create aneurysms. Then we use aortic ring cultures to screen our targets, the target pathways and the lead compounds. And in vivo studies, in case of medial artery calcification, we use chronic kidney disorder model, which has been, and since chronic kidney disorder, there is a lot of mineral imbalance going on, and it has been shown to be the direct cause for calcium deposits in the aorta and for the calcification of the arteries. Now, another part that we work on parallelly is that we develop nanoparticles for targeted therapeutics. We have a patented technology. We tag our nanoparticles with an antibody that specifically recognizes the damaged elastin, which is always the case, which precedes the arterial calcification. So by tagging our nanoparticles with this patented antibody, flexibzumab, we can target our nanoparticles specifically at the site of calcification and prevent the off-targeting side effects of the senotherapeutics or our target molecules. So, and then we study the effector pathways in context to SASP before and after the targeted drug delivery approach. So now, these are some of the results from my current research. So here I'm showing that by using the calcified artery, this aortic ring culture, we take the aorta from the healthy rat or mice. The aorta is sectioned, and the full length of aorta can be sectioned into 3 and 4 millimeter pieces, and it can be cultured individually, and different kind of pathological conditions can be simulated by this approach. So we used 2 conditions just to check whether senescence precedes arterial calcification or not. So in this case, we cultured the abdominal aorta, the 3 to 4 mm abdominal aorta in the control medium, complete medium, DMEM, and then we cultured another set of aorta in the high phosphate conditions, which are encountered in case of chronic kidney disorders. And we used the SA-beta-gal testing. We did the SA-beta-gal assay, and by doing this, we were clearly able to see that senescence precedes calcification. This is a short day culture, short day ring culture. We cultured the aortas for 5 days, and calcification occurs, the calcium deposits. Using this method, we have established that calcification occurs at a time point between 7 to 14 days. So we did a short term culture for 5 days, and we harvested the aorta for SA-beta-gal testing at a shorter time point to see if we are able to see any accumulation of the senescent cells. SA-beta-gal is the marker here that we tested for, and we clearly see that senescence precedes calcification in the aorta. And in case of chronic kidney, and when we moved, then we switched to our in vivo testing by using the chronic kidney disorder model in the rats. We tweaked this model with vitamin D and adenine diet at different stages and in different quantities to have a early and a late stage of the disease. And here we can see in the early stage, no calcium deposits. This is the elazarine red. So you see calcium deposits as bright red stain. So no calcium deposits are seen in the early stage, but the kidneys and the serum creatinine profile is significantly affected. So this is the early stage chronic, early stage kidney disorder, but the changes, the calcific changes are neither visible during the histopathology testing nor during the micro CT scan, but the senescence, the percentage of the senescent cells is increased. We are able to show here that senescence precedes calcification in vivo. So all the markers for the senescence like P21, P19, SA-beta-gal, they all are increased even in the early stage of the disease. Now, after having established that senescence precedes calcification in the in the medial artery calcification disorder, we have been working with the chelating agent. We have been working with this EDTA, and we have been making nanoparticles for this EDTA to reverse the calcification, because EDTA has the property to chelate these calcium ions and phosphorus ions. So when we tested these targeted therapeutics, we were able to see that it not only reverses, it not only chelates the calcium. This is before and after the treatment. The EDTA nanoparticles are able to remove the calcium from the arteries. They bring down the ossification markers, since the vascular smooth muscle cells, they change their phenotype from being smooth muscle cells to bone-like cells. So the osteocalcine, the RUNX2, these are all the ossification markers. These are all brought down by EDTA treatment. Survival time is considerably increased. And also the IL-6 and IL-1 beta concentration in the serum, that are the SASP markers, major constituents of the SASP. I would not say SASP markers, but the major constituents of the SASP, their concentration also decreases in the serum. So we wondered, what pathway is EDTA targeting other than just chelating the calcium ions, and is it doing anything intracellularly? So we also wondered whether it is able to decrease any senescence, since it is taking down IL-6, IL-1 beta in the aorta. The relative expression in the aorta, genetic expression, is decreased. Also, the MMP9 and MMP2, the marker, these are the other constituents of the SASP that go down. We saw that in a full-blown artery, in the late stage, or a fully blown medial artery calcification, this number of the senescent cells is also decreased. So we tested whether it is causing any kind of, apoptosis in the senescent cells, or in the aorta, or is it causing? So we tested for the apoptotic marker. We checked for caspase-3. But it does not decreases, it does not induces apoptosis. It rather brings down the caspase-3 expression. So it is definitely not acting as a senolytic. It is not a senolytic drug, but it brings down all the SASP markers like IL-6, 1 beta, MMPs, the senescence, the SA beta gal senescence. So then, what is EDTA doing, or how is it providing the environment by chelating the calcium around it? It's modifying the local environment so that it gives the opportunity for the cells to revert back to the healthy conditions. So for this, we moved to the immortalized human, our cell culture model. We use immortalized human aortic smooth muscle cells. We tested if caspase-3 is going down there by EDTA treatment. So we did 4 treatment conditions here. First was control. They were maintained, the cells were maintained in the complete media. In the second, the media was supplemented with a high, cells were maintained in a high phosphate condition. In the third, we used high phosphate condition along with EDTA, since EDTA can chelate the phosphate ions. And then we used it against the high phosphate conditions against ABT263, which is a known senolytic. And we tested what happens to the vascular smooth muscle cells and the caspase-3 expression in there. So we see that ABT, being a senolytic, it increases the caspase-3 expression, whereas EDTA does not. It is definitely not a senolytic. It does not causes apoptosis in the cells, whereas high PI conditions, they induce apoptosis by inducing caspase-3 expression. Then we moved back. So how to look for what is EDTA doing then, or what is the target pathway that we should be studying. So for this, we took leads from the existing literature, from the pioneers in the field of aging and cardiovascular research, and then we went back to the basics for the biomarker identification. And we narrowed down to the current target molecule and the pathway that we are studying in our lab. And this has also been marked as a potential source of SASP and in the senescent cells. I cannot reveal this target pathway right now, but we went on to see if high PI, if it is expressed in prior to the full-blown calcification. So we again did a short-term ring culture. We did a 5-day aortic ring culture model, and we were able to see that this particular pathway is upregulated. Is upregulated prior to the induction of calcification. And when we did a full-blown 14-day aortic ring culture, where we know that calcification has ensued, we saw a robust increase in its expression. And same way, we were able to see this target pathway being upregulated by high phosphate conditions in the cell culture model, which was unaffected by ABT263, but was being taken down by the EDTA treatment. Again, in the in vivo study, we were able to see that in a full-blown chronic kidney disorder model, when these were treated with the blank nanoparticles, which were not carrying any drug inside them, this target molecule was being expressed. Its genetic expression is being changed considerably, which is taken down by EDTA nanoparticles. And so we also did the effector molecules for this pathway, and we saw a decrease in the expression of in their expression as well, but that data I'm not showing here as of now. So what do we have from here? The current our current status in where we are right now is that mineral imbalance in chronic kidney disorder, it precipitates premature senescence. So it's definitely changing the proteome in the vascular smooth muscle cells in the arterial tissue, and that is what we will be looking in our future studies. Then, in context to medial artery calcification, senolytic approach is not a preferred approach because it causes heavy apoptosis. So therapy with Senomorphics could be a preferred or a potential way to move forward. And then Senomorphics affects it. This effect, it can be achieved by restoring the mineral imbalance. Since our targeted chelation therapy, it decreases SAS and has and potentially decreases paracrine senescence. So these are my findings from the current study in our lab, and we believe that this therapy has the potential to move forward to treat cardiovascular disorders. And with this, I would like to end my presentation and would welcome any questions from the viewers.